So good afternoon, everybody. It is an absolute pleasure to have Brian Munsky here. I've known Brian for about 11 years. He's a fabulous human being, a fabulous citizen, and an outstanding scientist. Brian has a very interesting academic path. He started out in aerospace engineering at Penn State, got both a bachelor's of science and a master's degree there. He moved on to earn a PhD in mechanical engineering at UCSB, Santa Barbara. And from there, he spent 5 years at Los Alamos, and that's where I got to know Brian. One of the interesting things about his time at Los Alamos is he was there for 3 years as a Feynman Distinguished Postdoctoral Fellow, which is one of the highest honors for postdocs at Los Alamos. Brian's work is fascinating. He has made pioneering discoveries in numerical methods to understand complex systems. Notably, he discovered what's called the finite state projection algorithm. And this enables predictive modeling for really complex biochemical systems. So an interesting fact about Brian, sorry to bring myself into this, but Brian and I together, we ran a 200-mile relay race together once. He's a great guy, and today he's going to be telling us about his stochastic modeling of single cell experiments. So go ahead and take it away, Brian. I'm happy to hear what you have to say.
Thank you, Ryan, for the introduction and for the invitation to speak here. Also, thanks for reminding me about that relay race, which was in Arizona, by the way, and that was super awesome and want to do that again. I'm going to be talking today about my lab's efforts to design optimal optical microscopy experiments to harvest single cell fluctuation information while rejecting measurement noise and image distortion effects. Oh, okay, here we go. I'm going to start off with a brief introduction talking about the origin of single cell variability or heterogeneity or noise, and the importance of including such noise in our models if we really want to understand what's going on in gene regulation phenomena. Then I'm going to talk about several different case studies where we're going to look at measuring and predicting the dynamics of single RNA transcription during fluctuating levels of stress. Then we're going to actually watch and model endogenous RNA polymerase II, the recruitment, phosphorylation, and then the eventual transcriptional activity of those polymerases in living cells. And then we're going to talk about observing and simulating canonical and non-canonical translation dynamics, again, looking at a single RNA molecule translating as it's diffusing inside the cytoplasm of a cell. In part two, we're going to switch gears. We're going to talk a little bit more about using mathematical models to improve the designs of single cell experiments so we get the most bang for our buck. So let's start off with a very brief introduction to the central dogma of molecular biology. What we're looking here is the thing that you learn in elementary school, that genes are transcribed to form messenger RNAs, and messenger RNAs are translated to form proteins, and it's the proteins that do all the real jobs inside the cell. Now, this is a really stochastic and random process, starting really at this gene activation and deactivation step. Most of our cells are only going to have one or two copies of important genes, and those genes may be on, in which case they can be transcribed, or they may be off. And the timing and the situations under which they switch between being off and on are very unpredictable because they really do involve a single molecule hitting the right conformation so that it'll be active. This results in a lot of variability into which genes are active in which cells, and that is inherited in the RNA distributions, as we're going to see, as well as in the protein levels. So the result is that genetically identical cells in the exact same environments, with the exact same genomes, the exact same initial conditions, can result in stochastic randomness that will result in spatial and temporal fluctuations, either within a single individual cell or among many cells in a population that all derived from the same ancestor. Our goal in our lab is to measure, model, and try to predict every single stage of these fluctuations so that later on we can go in and genetically modify or change the environmental conditions so we can get these fluctuations to do what we want them to do, at least in a statistical sense. Okay, so let's bring in some modeling so we can understand how that can help us to understand gene regulation. Here's again a cartoon now of a bursting gene expression model where a gene may be off or it may be on. It can go from off to on at some rate k on, And it can go from on to off at some rate K off. When it's in the on state, it may transcribe messenger RNA at some rate Km, and those can decay in a forced order decay process at some rate Gn. Now, this is a model, it's known as the random telegraph model or bursting gene expression model, and it's been analyzed like crazy. Here's a bunch of papers that looked at that model in different contexts. This model has been solved in many, many different ways, and we know a lot about its solutions. So what I'm showing here are steady state solutions. We can ask what's the fraction of cells that are going to be in on state at steady state. It's just given by this little ratio. We can ask what's the mean level of RNA that are produced at steady state under this model. Again, we can compute that from those parameters. We can also compute what's the variability or the variance in the steady state distribution for this model. We can get the variance, and if we divide that by the mean, the variance divided the mean is known as the Fano factor. And again, it's a nice, simple, computable expression that describes this process in terms of its parameters and the results of this process. So what we can use this for is to see if we had measured some of these properties, the mean and variance, what can we learn about the parameters of the gene regulation mechanism? So what I'm showing over here in white lines are lines of constant mean expression. In case this one corresponds to 2 molecules of RNA on average, this one to 25 molecules of RNA on average, as a function of the different parameters, the rate at which it turns off or the rate at which it turns on. Now, if we were to measure this biological process using techniques like PCR, which give us a bulk measurement of a lot of cells, the best we can hope to measure is this average expression level. We'd know that there's 2 molecules of RNA on average per cell. But that would tell us only that we're along this line somewhere, but we wouldn't be able to uniquely determine the parameters from that particular measurement, because anywhere on this line would be equally likely. Now, if we measure the variability as well, we can get a sense of, is it highly variable, like this red region for the Fano factor, which is a highly variable population, or is it relatively homogeneous, like the blue region, which has relatively little variability? If we were to do this experiment again, but now measure the amount of variability, and we find that it's in the yellow region, a mid-level of Fano factor, then we would know that we're somewhere along the yellow region due to the variability, but we're somewhere along this white line due to the measurements of the mean, and that would tell us that we're right around this sweet spot in parameter space. We'd be able to almost uniquely identify these parameters based upon those measurements of variability and average expression. Now, I can extend this a little bit further because the beauty of this model is it's used to describe gene regulation. How are genes being activated or induced, or how are they being deactivated or repressed? So if we can look at this, we can ask that there's 2 different ways we can activate this system from a mean of, say, 2 molecules up to a mean of, say, 25 molecules. The 2, according to this diagram, is the first one we could do is we could decrease the rate at which it switches off. This would correspond to something like a kinase or that transcription factor that somehow modifies the chromatin to stabilize it in an on state so that it's going to keep producing RNA for longer before it randomly switches off. If we do that, we go from here to here. The other possibility in this model is that we increase the rate at which things turn on. This would correspond to adding a transcription factor that binds to the promoter to activate it. That would take us from this point to here. We're increasing the rate at which it turns on. Now, although both mechanisms take us from 2 to 25 molecules on average, and the bulk expression is the same for both mechanisms, it's highly different as to the effect on the variability. In one case, in the first case, it resulted in a net increase in variability, and we ended up with a distribution that looked like this bimodal, very heterogeneous distribution here. And in the other case, it actually decreased the amount of noise that we saw in our population, resulting in this distribution here, which looks a lot like a Poisson distribution. So by keeping track of how the variability and the average expression change as a function of condition or over time during a dynamic response gives us a lot of insight as to what the parameters might be, but also what might be the potential mechanisms that are giving rise to these regulation phenomena. Now, in addition to steady state processes like I was describing before, we can also simulate this model quite easily using, say, a Gillespie stochastic simulation algorithm. And we can look at results of these kinds of trajectories, or if we did this experimentally, as we'll see in a little bit, we can measure trajectories of this nature by looking at a single cell. If we were in this particular example, if we were to measure the times at which it's spent in the off state, and we look at the average length of these times in the off state, well, that's directly related or inversely proportional to the rate at which it turns back on. Similarly, we could look at the average length of time it spends in the on state, and that would tell us information about the rate at which it turns off. There are other things we can start to pull out from this system. One example would be the amount of RNA that are created in a burst, the total amount that are created in a burst, and that would be related to the parameter of the production rate divided by the rate at which it turns off. We could also look at the average height that this thing achieves when it's in the middle of a burst, and that could be related to the production rate divided by the degradation rate that the RNA is seeing. We could also look at the time it takes from when it switches off to when the RNA goes to 0 and look at the half-life of that decay process. And the half-life of that decay process would give us some information about the degradation rates. So there's lots of different potential ways we might be able to measure these parameters. And we want to get at a more rigorous ways to choose which of these experiments would be best suited to identify parameters of a given model. One of the mathematical techniques that we're going to use is the finite state projection approach that Ryan briefly mentioned during the introduction. This is an approach that we developed many years ago now that allows us to solve for the non-stationary distributions of time inhomogeneous master equations, or Markov processes. The basic idea is that at any point in time, we're going to define the state of the system as an integer population vector, 10 molecules of species 1, and 15 molecules of species 2 in this example. Reactions are transitions from one state to another state. This was an increase in 1 molecule of species 1, a degradation of 1 molecule of species 2, another increase in species 1. Now, these are random transitions. Instead of going right, then down, then right, we could have gone in any other direction as well. And we could have reached other states. The problem becomes, given that we know that we started at some initial condition, or perhaps we know that we started at the stationary distribution in some unperturbed setting before we added environmental stimulus. Given that we know that initial distribution, we want to ask ourselves, or want to compute, what is the probability of being in some other state, like say over here, at some later time t? Or what's the probability in being in this row versus this row versus this row at some later time t? And that's the t at which we're going to do our experiment. To solve this problem, what we do is we formulate a large set of linear ordinary differential equations that are known as the chemical master equation. The chemical master equation is actually very easy to write for a given chemical reaction, although it's potentially infinite dimensional because there could be an infinite number of points on this lattice that I drew. The master equation describes the time rate of change of the probability of a given state. Let's say that state for right now. It's telling us how the probability of that state is going to change as a function of time. It has a negative component corresponding to the flow of probability that leaves that state and goes somewhere else. And it has a positive component corresponding to probability that starts at one of the neighboring states, let's say here, here, or here, and then flow of probability into the state. This is a transport problem. It's a transport of probability mass that we're looking at. So we have to write one of these equations for every single possible state on the lattice. And as a result of that, the master equation becomes this very large linear ordinary differential equation. It may be time inhomogeneous if any of the parameters in this matrix are functions of time. We can write this master equation very easily. Well, at least we can write finite portions of it very easily, but we can't solve it exactly because it's infinite dimensional. The finite state projection approach was basically a way to take this and create a new fake master equation that has a finite number of states. And we did it in a very specific way, where we reproduce everything inside the region that we want to keep exactly as it was, and everything else we shove into an absorbing sink state. The result of this is a finite dimensional master equation, in this case, a 7-dimensional ordinary differential equation, which is really easy for us to solve numerically. Now, there's three very important statements we can make between the relationship of this master equation and this one over here. The first one is that the solution of this is a guaranteed lower bound on the true solution of the master equation. So this is strictly less than or equal then. And furthermore, if we were to take the true solution and select and subtract our approximate solution for all of the points in this region in cyan, and then if we were to subtract 0 from everything else, and note that this subtraction is always going to result in a positive number based upon this property. If we were to add those all up, it would be exactly equal to some quantity epsilon, which is a function of time. And this epsilon is exactly the solution to this ordinary differential equation. So we know what the exact error is for any approximation we make. And then the third important property is that if I were to change this projection by adding some new states, if I were to go to a projection J1, which has 6, to some J2, which has now 7 states included, but those 7 states include the original 6 states, then I'm guaranteed that the error for my new projection, epsilon J2, is guaranteed to be less than or equal to the error for the original projection. And this means I have monotonically decreasing error. My error can only get better, it can never get worse. And a result of that gives us a chance to derive a much more important property, which is how to use this thing in the comparison of a model to real experimental data. We can ask, what is the likelihood of experimental data D given some parameters, they're represented by theta, and based upon these 3 properties, or based upon these 2 properties, this one right here, we are guaranteed a lower bound in the log likelihood of the data given the model from this. Furthermore, these 2 properties allow us to compute relatively easily compute an upper bound on the likelihood of the data given the model. And so now we've got a monotonically converging lower bound and a monotonically converging upper bound, one converging from below and one converging from above, and they monotonically converge to the exact solution for the likelihood of the data given the model. So this gives a very precise way to estimate what is the likelihood of the data given a model, and we can use this to compare it to 2 different parameter combinations or 2 different models and ask which one is better able to reproduce experimental data in a statistical sense. Okay, so now we're going to have these tools. Let's start to talk a little bit about some of the experiments we can do, and we'll show how we can fit some models. Now I'm going to go through these relatively briefly. If you'd like more details, I'm going to give you the contact information for all the postdocs and graduate students who have done all this work, and I'd be happy to answer a lot of questions as you have them. Let's start with measuring and predicting the dynamics of single RNA transcription processes during fluctuating levels of stress. So here we're going to start with an experimental technique known as single molecule fluorescence in situ hybridization. I suspect many of you have seen this before. The idea of single molecule FISH is that you have a long messenger RNA, you know the sequence of that RNA, you design small DNA oligo probes. This is a probe that binds at exactly that region of that RNA. This is another probe that binds here, another probe that binds here. Each of these probes is conjugated to a fluorescent dye molecule, such that we've got 20 or 30 or 40 of these probes that are all going to bind to a single molecule of this RNA. When looked at under a microscope, a diffraction limited microscope, this entire messenger RNA is about the size of a pixel or 2, and that pixel or 2 is going to correspond to a single molecule of RNA. But it's a very bright spot because there's so many dye molecules all colocalized at that single point. There could be more than one RNA. There'd be a bunch of these RNA, and we will image these in Z-stacks to get 3D resolution to see where all the spots are inside the cell, both how many there are and where they are. This is an actual yeast cell now that we're showing here, where it's about 5 microns in diameter, and we can just go in here and count the spots. 1, 2, there might be 2 there, so 4, 5, 6, etc. And so we can count how many spots are in this individual yeast cell. And we can do this for thousands of cells at different points in time during a dynamic response or under different environmental conditions. Here, in particular, we're looking at a stress response gene where we gave a molecular signal at time t equals 0. And after 2 to 4 minutes, we see RNA starting to be transcribed inside the nuclei of some of these cells. So that's a site of nascent transcription. We're actually watching the birth of RNA there in that fixed cell. We caught it in situ, in the situation of that those RNA being born in that location. We can measure these for lots of cells, thousands of cells, and by counting how many have 0 molecules of RNA, how many have 1, how many have 2, et cetera, all the way up to 80, we can get a probability distribution or a histogram for how many cells have every different possible number of RNA. Now, what's really cool about this process and looking at these systems, this is a highly stochastic process in that all of these cells are behaving very differently from each other. And there's no chance that we're ever going to predict exactly what a given cell is going to do, nor is there any point in predicting what a given cell is going to do. However, if we look at the probability distributions, the histograms of this response, we find there's a very large amount of reproducibility. In blue and green are 2 completely separate experiments that Gregor did when he was a postdoc. They were separated by the entire year with completely independent yeast populations. And yet you can see that the reproducibility between one replica in red and another replica in blue is nearly perfect. This provides this nicely reproducible set of data that we can now fit models to. Here's an example of what a model might look like. It's very similar to that bursting gene expression model that I started the presentation with. The only difference is it now has 4 states for the bursting gene expression model, and it also tracks the spatial location of messenger RNA, whether they're in the nucleus or the cytoplasm. We have some stress that activates a kinase signal, which is shown here under 2 different levels of environmental inputs. We can fit a model to that very nicely. So we've got a nice controllable input signal. And then under that, we can measure in the colors and different replicas in the colors, and we can fit a stochastic model using the finite state projection approach I showed earlier. We can fit a model to that to fully reproduce the distributions of the RNA per cell as a function of time with 1 to 5 minute resolution. And we can do this for multiple genes and in different genetic and environmental circumstances. We can also measure the joint probability distribution for how many are in the cytoplasm versus the nucleus, and the model can reproduce that quantitatively as well. After we fit a bunch of the stochastic, spatial, and temporal data, we can then use a fixed model, and we could predict for the probability or the intensity of these transcription sites, again, as a function of time, a function of gene, and a function of the environmental circumstance. So that gives us a nice quantitative predictions that we can then test through experiments. This is yeast cells, just very briefly. We did something very similar recently in human cells. Here we're looking at human cells, IL-1 beta and TNF alpha in red and green respectively. These are response genes that are responding to bacterial infections. We simulated this by introducing bacteria LPS into the cells, which are activating these gene expression. Once again, we found a very simple bursting gene expression model. In this case, it had 3 states for TNF alpha and 3 states for IL-1 beta, but the transitions between these states depend upon different kinases or transcription factors in this case, and the levels of those transcription factors depend upon the amount of stress, the LPS signal, as well as how much drug different types of drug that we give to the systems to reduce inflammation. We can find a single consistent model that can now once again reproduce the probability distributions for the activity of the IL-1 beta and the TNF alpha as a function of time and under different drug combinations that we're just showing the wild type here, but under different drugs as well. So we get a nice reproducible stochastic model that can reproduce these single cell snapshot distributions. We can go a little bit further, and we can now, with more modern experiments beyond single molecule fish, we can actually do a lot of what I just described in those fixed fish cells, but we could do it with live cells and actually watch what's going on. So first we're going to watch, and we're going to develop models of RNA polymerase 2 or RNAP2, the recruitment, the phosphorylation, and the activity of that as it's activating gene transcription. So here we're showing a movie, and what we're watching here in red is the activity of a transcription site inside the nucleus of this human-derived cell. We can see it bursting on and off as nascent RNA are being produced or destroyed at, or produced or released at that site of nascent transcription. The way we do this is very similar to single molecule fish, although it's a live cell assay. We're using MS2 stem loops on the messenger RNA. They're genetically encoded into that. They're recognized by this red MCP coat protein. So as the polymerase moves from left to right, as the nascent RNA elongates, it gets labeled by these red dye markers, allowing us to track it and also allowing us to see how much transcription is going on at that site. Now, we extended this technique using fluorescently labeled fragmented antibodies, or FABs. These fragmented antibodies allow us to quantify the amount of RNA polymerase in blue that's being recruited to the site of transcription. As these things are labeled in blue, we see them cluster around this transcription site. We also add a second FAB, which will bind specifically to phosphorylated RNA polymerase II. And this phosphorylation step is a necessary step for the activation of transcriptional activity of the polymerase. So what we see now is the polymerase coming into the transcription site very rapidly in bursts. It gets phosphorylated very quickly, so it goes from being just blue to being blue and green. And then a short time later, we see the RNA being transcribed from that same transcription site in that same co-localized location at the site of the gene. We can take these dynamics and we could try to get some of the statistics from them. Now, if we were to watch this trajectory for a different cell, we'd notice that every single trajectory is different from all the rest. It's because of the intrinsic variability. We're never going to expect a model to fully reproduce any one of these trajectories, but we can ask that our model is capable of matching the key statistics of these trajectories. And we're going to ask the model matches the statistics in two ways. First, it should be able to reproduce the probability distributions of intensity of the different colors. For example, here is a model shown in green and experimental data shown in magenta for the probability for the number of nascent transcripts per transcription sites. And we can see that we can reproduce that very, very well. I'll show you the model in the next slide. We can also ask the model is able to capture the correlation statistics. In this case, we can look at the autocorrelation, that is how correlated is a signal, let's say the RNA intensity at time t, how correlated is it with itself at a later time t plus tau. That gives us this autocorrelation. This kind of gives us the characteristic time of this process. How quickly is the transcription occurring? How quickly are these RNA being processed before they're released out into the rest of the nucleus? In addition to autocorrelation of the signal, we can also ask for the cross-correlation of the signal. What is the correlation of SER5 at time t with the mRNA at some later time t plus tau? This allows us again to see, if there's any kind of time delay. In this case, there is a time delay between when the SER5 shows up here and when we see the RNA signal in red a little bit later. This asymmetry of the peak tells us how long does it take for the polymerase to go from the place where it starts to where we actually can see it based upon the red probes that we engineered into the system. So a good model should be able to fit all these statistics very well. But when starting this process, we don't know exactly what that model is going to be. In order to get at that, we had to hypothesize lots of potential different models. Here's an example of 6 of those models, listed from the top, which is the simplest model, down to the bottom, which is the most complicated model, showing all the parameters as well as how well it fit, the log likelihood, and then a criteria of Bayesian information criteria to discriminate between when model's too simple or too complex. The first 2 models that lack a separate mechanism for RNA recruitment and transcriptional escape, we found that they were too simple. They just couldn't fit to the data very well. The last 3 models fit to the data extremely well, but we found that those models had too many free parameters. They fit to the data very well, but those parameters were so uncertain that we couldn't really identify unique parameter combinations for things like extra phosphorylation steps or distinct RNA elongation processing steps or other bells and whistles that we were adding to this model. Based upon the amount of experimental data that we had available to us, we can select this Goldilocks model in the middle that minimizes the Bayesian information criteria. It also is optimized, selected as optimal based upon the Ikaiki's information criteria. And this is the model we go with. So let's zoom in on that model. So this is a relatively simple model. It consists of 4 reactions and 5 parameters. The reactions are 1, 2, 3, 4. The first reaction corresponds to bursts of recruitment of the polymerase to the site of transcription. They occur with a burst size beta and at a frequency of omega. When they're there, they can either escape to result in active elongation of the messenger RNA, or they can abort, resulting in their lost or unsuccessful attempt to create RNA. Once they've escaped, they're going to spend some time at the site of transcription, which is taking for account both the elongation of the messenger RNA as it polymerizes, as well as a processing of the RNA before it's released into the rest of the nucleus. And that has a rate case C. We used Markov chain Monte Carlo, specifically the Metropolis Hastings algorithm, in order to identify both the parameters as well as the confidence intervals on those parameters for all these different parameters. Once we have that model, we find that it and its uncertainty over parameter space does a very, very good job of reproducing all of the quantitative observations we have. We can reproduce the probability distribution for the intensity of the CTD signal at steady state in our cells. Same thing for the SERA5 signal. In real numbers, we can reproduce the probability distribution for the number of nascent RNA. We can also get the autocorrelations for the CTD signal, the SERA5 signal, as well as for the messenger RNA. So each of the different signals autocorrelations, and we can get the cross correlation of any 2 pair of these signals. From those, we can look at the peak of that, and we can use that to identify what are the characteristic times it takes for the polymerase to move from one side to the other side of the messenger RNA, or of the gene specifically. We could also take this model and see if we can learn some more insight on how this model is perturbed when we stress the system. For example, we could look at different transcriptional inhibitors. The first one we looked at was triptylide, or TPL. This is a drug that works by prohibiting the DNA from opening up, which therefore prevents the recruitment of the polymerases to the transcription site, and it will therefore eliminate the nascent transcription. In our model, that corresponds to just repressing this particular reaction right there. And as a result of that repression, the transcription goes to, the transcription goes to 0, as well as the clusters of the polymerases go to 0. And that's exactly what we see in the experimental setup as well. The second drug, THC1, works by inhibiting the SERA5 phosphorylation. It's basically blocking the escape part of this dynamical process. This blocks the RNA polymerase from escaping, and therefore there won't be any transcription as well. And over time, this is going to result in much less transcription, just like we see in experiments. However, this does not block the recruitment of the polymerase, so we still get fluctuations of the polymerase at the transcription site. They're just not actively resulting in RNA. The third experiment, flavopiridol, prevents RNA elongation, so it stops the escape, but it's also stopping things from releasing as well, although be it to a slightly lesser extent. So there's not going to be any new transcription, but the old transcription is going to take a while to go away. And looking at that model, we see that it does slowly reduce the amount of RNA at the transcription site, although it's much more variable than in either of the other 2 circumstances. And it still does allow for a pretty large burst and a lot of noise and a small decrease in the overall amount of the polymerases in the cluster. Okay, so that's transcription. Now we've now looked at half of the central dogma of molecular biology, and I'd like to now look at the second half, the other half of the central dogma of molecular biology. Let's look at translation. So let's see how we can observe and simulate both canonical, that's the regular translation that most of our genes are doing, as well as non-canonical translation that viruses do when they infect our cells. So let's start off with a slight modification of that experimental technique I just talked about. Well, in fact, we're going to use the same technique. We're going to use the MCP-MS2 labeling. There's a spot you can watch a messenger RNA diffusing in this cell. You can see it moving it around. But in addition to that technique, we're also going to clone in small peptide regions in the translated protein shown here in white. So those small peptide regions are going to form a secondary and tertiary structure that are now recognized by, again, some fragmented antibodies. Those fluorescently labeled fragmented antibodies are going to bind to the protein as it's being created in real time. So as this, here's a good example right there. You can see the green. The green is being, puffs of green are being released from this messenger RNA as it's moving inside the cytoplasm of this cell. Using this technique, we can again get a lot of experimental data. I'm going to show you in a little bit. We're going to develop a pretty simple model known as a totally asymmetric simple exclusion model, or TASEP model, which consists of only three important mechanisms. The first mechanism is it can initiate, ribosome can bind to the RNA at some rate. Then the ribosomes can move from left to right without passing each other. They can't pass each other. And when they get to the end, they finish. And once they've passed to the region of the labeled probes, they're going to show up as green as they do in our experimental setups. They said this model only has two free parameters, the initiation rate at which it starts, and then the average elongation rate over the entire genome. Using the average elongation rate and having knowledge of the codon usage of the human genome, or the tRNA prevalence inside human cells, we can convert that to a codon-dependent translation rate at every single point along the gene, or along the RNA. But just specifying these two parameters, we can reproduce the number of nascent proteins per messenger RNA, as well as the autocorrelation of a signal with itself. That's the intensity or the time scale of the intensity memory loss for different RNA constructs. Here we're showing that the same model and same set of parameters are able to reproduce the distribution of nascent protein per RNA for a very short gene of 128 codons, as well as for a longer gene of 375 codons. We see that the longer gene has a slightly brighter signal. This distribution shifted a little bit to the right. And we also see that the longer gene lasts a little bit longer. This decorrelation time also shifts a little bit to the right. It's persisting for a little bit longer. Now we can take this canonical translation experiment and model, the TASEP model, and now adapt it for use to understand viral translation dynamics. And that's what I'm showing here. This is just an image of HIV virus particle. HIV and other viruses use this technique of frame shift stimulatory sequence, or FSS, to reduce their genome size. What a FSS is, it's this hairpin structure that shows up in the RNA of a viral translation, of viral messenger RNA. These ribosomes are going to cause these sequences, the FSS, is going to cause a ribosome to get stuck there. In natural normal translation, the canonical translation we just talked about, as a ribosome chugs along from left to right, from the 5' to the 3' end of a messenger RNA, it's reading words in letters of, and it's reading words with 3 letters apiece, the codons, and it's getting an amino acid sequence of H, G, F, L, etc., and so forth. Each set of 3 letters gives us a new amino acids that's being associated onto this elongating protein. If a ribosome gets stuck behind this stem loop, where its center, the coding region, the actual work that's being done by the ribosome, is stuck in this poly-U region, it can get confused. It can wiggle it around. It might get knocked off track. And the first 3 amino acids would be the same as before, HGF. But if it gets knocked back 1 nucleotide, instead of reading UUA and coming up with an L, it reads UUU and comes up with another F. And then if it's released, it continues on this new track and it's creating a completely different protein. For viruses, these proteins, the green protein and the part green, part blue protein, may actually have functional significance. And by doing this, the virus was able to pack two proteins on the same coding region. Very useful for it, so it has a better chance of evading our host immune response. In order to look at this experimentally, Ken Lyon, student Tim Stasevich's group, what he did is he created two sets of those fluorescent fragmented antibody probes, one in green that's going to be on the 0 frame, another in blue that's going to be in the -1 frame. So just by looking at this thing, we're going to know if it's just green, it's canonical translation, and if it's blue, it's frame shifted translation. We also developed a model that is going to have the same initiation rates as before. It's going to have the same elongation rates, but there's going to be a probability of it switching from an open stem loop where there is no stem loop to a closed stem loop, which is going to result in both a pause and the shifting over to the -1 frame. Here's what it looks like experimentally. We can see there's multiple different types of spots. Here's a spot in red. It's this set of crops. It's just red. There's no translation either in the 0 frame or the -1 frame. Should note that this is diffusing very rapidly because it's a very small complex. Ribosomes are quite large. This other spot, number 2, we see it has the messenger RNA. It's also translating in the green frame, but not in the -1 frame. This one's moving a little slower than number 1. And then this third spot, which is moving very, very slow, is actually translating both in the green frame, the canonical, as well as in the -1 non-canonical frame shifted frame. And it's moving slowly because it's got so many ribosomes all kind of lumped up on top of it. We can develop a model, again, it's the TASEP model, and we can produce all of the quantitative information that we can glean from those experiments. We can ask our model to reproduce the survival time of the frame shifted spots. We can ask the model to reproduce the loading of the ribosomes per RNA in each of the frames, whether it's a 0 frame only spot, a -1 frame only spot, or if it's a both frame spot, we can reproduce the amount of 0 frame product or -1 frame product in those spots. We could also ask that our model is able to reproduce the fraction of mRNA that are translating either in the 0 frame, in both frames, or just in the -1 frame, as well as the fraction that aren't translating at all. We can also ask that our model can reproduce the ratio of the 0 frame and 1 frame protein products. It's about 4%. About 4% of the proteins created from this construct are in the -1 frame, and 96% are in the 0 canonical frame. We can also look at the effects of stress and drug perturbations. Drug perturbation we can look at is Harringtonian, which stops translation initiation. And we can look at it for 2 different genetic constructs. The first construct on the left here is showing where the 0 frame is going to show up in green and the -1 frame is going to show up in blue. And the 2 different sets of lines correspond to whether or not they have blue. If they have blue, they're going to be represented in the circles. And if they do not have blue, they're represented in the green triangles. For this particular construct, there's not a whole lot of difference between these 2 runoff dynamics. You give it stress there, it all goes away after about 800 seconds. In this construct, our model predicts that, so in this construct, we put an set of orange probes upstream from the frame shift site. We are going to ignore these probes, and then we're going to downstream for the frame shifted system, we're going to use blue. Now, once again, we could separate all of our spots amongst those that have blue and those do not have blue. They're all going to have orange, which allows us to track them. So these are the spots that have blue, and these are the spots that do not have blue. And this is the prediction of our model and for each of those particular constructs. Now what happens is this frame shift site, when it forms its secondary structure, the ribosomes can't get past and they get blocked. So if that structure is not there, it goes through this way and everything disappears quite quickly. It takes longer than it did over here simply because there's a long region here. So this construct is longer than this construct. However, when the frame shift site is there, when there is a hairpin, the ribosomes cannot get past they get stuck, they start to pile up in a traffic jam behind the frameshift site until it eventually releases. Because it takes time for it to release, and because they're going to bump into each other to clear after the traffic jam goes away, those that go through this pathway and turn blue take way longer, up to an hour before they're actually going to clear after the stress. And so the model is able to help us both understand that dynamic, and we knew to expect to see it, so we could actually design this to show that that would happen. We can also use this to look at other aberrant non-canonical translation process, but given the time, I'm going to have to skip that. I want to get to the next part of the talk. Okay, was there a question? No. Okay, so conclusion parts 1. The central dogma is very noisy, very stochastic, and RNA proteins are created in discrete bursts. That bursting gene expression model is very, very useful. Transcription and translation bursts can be measured in real time at single molecule resolution and in living cells. Simple discrete stochastic models like the ones I showed, which are actually quite easy to analyze, are sufficient to quantitatively reproduce every observation we have from these advanced experiments. By testing multiple models in different stress or drug response conditions, it is possible to gain insight into which bursting mechanisms are most likely to account for the observations that we're making. But I want to switch gears here and ask, can we do better with those types of experiments? Single cell experiments like those I was just showing are extremely powerful. They're really cutting edge. We're very excited by them, but there's some problems, some drawbacks. First, there's a lot of experiment design considerations. How many cells do you need to measure? What should be your sampling period? What fluorophores should you be using? Where should you be placing them on these genetic constructs? What inducer or drug concentrations should we be using? When should we be measuring the responses? There's also a lot of experimental error considerations we need to take into account. What is the resolution of our microscope? How good is our image processing? How well can we segment or detect spots? What about photobleaching? What about autofluorescence of these cells? What about delays and how long it takes the drug to get to the DNA or RNA before it shuts things down? We want to get as much insight as possible out of these experiments, which means we need to choose the experiments that are most likely to maximize the information about the processes that we actually care about. For that, we use a technique called the Fisher information matrix from statistics. The Fisher information matrix is a very useful tool that quantifies the amount of information that we can expect to get from observed random variable. For example, the observation of how many RNA are in a cell using single molecule FISH. We can write the Fisher information in terms of that log likelihood I showed earlier, the log likelihood of the data given the parameters. We can look at the gradient of that with respect to parameters, transpose times itself, and take the expectation of that over all possible consistent experimental observations. This is something we can compute very easily now using the finite state projection approach. It's just an algorithm. You have the FSP solution, which is this. You can take its derivative with respect to parameters very easily. And then using that, you just plug it into this equation. You've got the Fisher information matrix. The Fisher information matrix is really useful because it provides an asymptotic estimate for what will be the uncertainty of a maximum likely estimator. So the idea is if theta star is the true parameter set and theta hat is what you estimate by doing an experiment and trying to identify what theta is, then the difference between these two converges in distribution to a normal distribution. And that's an unbiased normal distribution with a mean of 0. And a covariance matrix is the inverse of the Fisher information matrix. So in other words, the Fisher information matrix or its inverse should be the covariance matrix for our error in our experimental measurements, or our maximum likelihood estimators. By looking at the eigenvalues and eigenvectors of the Fisher information matrix, it can tell us what will be the confidence intervals of our maximum likelihood estimator. The really poorly identified direction is going to correspond to the smallest eigenvalue of the Fisher information. And that direction is going to be the eigenvector corresponding to the smallest eigenvalue. The distance of the best identified direction in parameter space is going to be related to the largest eigenvalue, in fact, actually 1 over its square root. And its direction is going to be related to the eigenvector for the Fisher information. We can verify that this is actually working by generating simulated data from a model estimating parameters from those different simulated data sets, make a scatter plot of those estimates, and then use that to compute what the confidence interval was from that MLE sampling. That's shown here in the black line. And we see that it exactly matches to what we expected it to be based upon the Fisher information matrix that we just computed directly from the model. Now, this takes about a 100th of a second to compute, whereas this Monte Carlo sampling to verify that it is actually working takes several weeks to do. So we have a very fast and efficient way of estimating how good our estimate's going to be given a model, so long as we have a model or we have some prior of that model. We're quickly running out of time, so let's see how we actually can use this. Suppose I had two potential experiments, whatever different experiments we've defined, and we can compute the Fisher information for both. The Fisher information for experiment 1, taking its inverse and looking at its covariance matrix, tells us we're going to get this much uncertainty in our parameter estimation. If we do it for experiment 2, we find that there's a lot less uncertainty. In terms of metrics of the Fisher information, this means that the metric, the size of Fisher information matrix for experiment 2 is larger than that in experiment 1. Which metric we use, whether we're using like the volume of the ellipse or we're using d-optimality, there's lots of different metrics we can use. Each one is chosen for a very specific problem. But in general, if the metric of experiment 2 is bigger than that of experiment 1, then experiment 2 is a better experiment than experiment 1. And you can use that to design which experiment to do. A more concrete example of that is, again, going to our bursting gene expression model. We're going to take single molecule fish data, but we're going to take it at different time points. We're going to take 3 data sets separated by a sampling period delta t. We can either do a slow sample where they're spread pretty far, or fast sampling where they're pretty close to each other. As a function of the sampling period, the blue line and the orange line, which is the validation via maximum likelihood estimators, shows us how much we can learn by doing samples at different rates. And there is an optimal experiment design right here, which is actually a very experimentally realizable experiment design. That tells us what should be the optimal experiment. The other 2 colors, you could ignore those. Those basically correspond to previous Fisher information approximations, which turn out to not be very accurate. And experiments designed based upon, say, this one in particular, which is based upon ordinary differential equation analyses, results in an experimental design that's actually really terrible. So using this for experiment design actually is worse than just intuitively guessing a random experiment. It overestimates the amount of information, and it makes you think you know more than you do after doing the experiment. But the one that we showed that this is actually correct, and we do it for we could show for different analytical solutions that it indeed gives you the exact correct solution. Okay, so this allows us to do experiment design. Here's an experimental validation of that using real experimental data. We're going to take that yeast experiment that I was talking about toward the very beginning, and we're going to assume that there's some unknown sodium chloride concentration, some salt concentration. We don't know what it is, but we want to estimate what it is from our experiments. We can do one of these 7 experiments. We tried lots of other ones, but these are the ones that we just randomly generate. The one at the top is one that was found via Fisher information to be optimal. We take those different experiments, we compute the Fisher information by considering the model that had already been identified in previous literature. Plug that in, solve for the Fisher information, and we ask what will be the uncertainty, the expected uncertainty in the signal duration, the Higgs signal duration. That's the signal duration of this part of the model. And we want to know which of these experiments will minimize that uncertainty. Then we take those same experimental designs and we collect experimental data or resample experimental data according to those designs with 75 cells in every single total population. We run those with maximum likelihood and try to infer what's the actual maximum likelihood error in the sodium chloride. So we're designing our model to maximize information about this, and we're going to compare it to how well it maximizes information about something that's closely related, but not directly related to it. Doing this experiment, or this thought experiment, we find that the Fisher information, which gives us the dark colored lines, as a function of all the different experimental design matches very well to the actual information taken from the experiments. So we're able to select which experiment designs are likely to identify the thing we're trying to get and which are not. The same thing over here. There's one exception. I'm happy to talk about the exception afterwards. Okay, the last thing I want to talk about today is going to a more realistic situation, even more realistic than that. We know that all experiments are flawed. No labeling strategy, no microscope, no image processing tool, no matter how much we are super proud of what we're doing, none of them are perfect. All measurements are noisy, all data is distorted. It's all an approximation of what's actually going on in the real world. So if this is a real cell, these are images that one of my students took a week or so ago. If this is a real cell, maybe your distorted image might look like this. If this were the true probability distribution, of course, we can't really get that, but if there were the true probability distribution, then maybe your distorted probability distribution for how many spots per cell might look like this. If you don't account for that variation, that distortion, if this were the true parameters combination for two parameters of interest, if you didn't account for that, you're going to get distorted estimates. They'd be biased away from the true parameter combinations. We can account for this by including a calibration step, a probabilistic calibration step, where the distortion of the data is modeled by a Markov kernel, distortion kernel. This is something that's a property of the microscope or the process that we're using to collect the data. This tells us that the true distribution is filtered through this Markov kernel to give us an observed probability distribution. If you account for that, and you can count for that both in your likelihood estimator as well as in your Fisher information. To count for it in the Fisher information, you just plug it right in there. You go from px to py. This was the sensitivity. It's the same linear operator. And after a little bit of math, it's relatively easy to do in a computer, you get what the new information would be. By accounting for that, you can bring your estimates back to the true parameter space. And the new Fisher information matrix matches very nicely to what you actually get in the spread of the maximum likelihood estimators. Okay, so let's finish up with our the application of that. Suppose you had 3 different experiments you could do. You had perfect single molecule fish. You had a lossy fish where you just miss half of the spots at random. And you have flow cytometry, which is a much faster but a lot less accurate measurement process. The kernel in this case would be an identity matrix. There's no loss of information. The kernel here is this kind of blurring. It kind of it's correlated, but it's blurred. Here, it's blurring quite a lot. After accounting for these, we can ask what would be the magnitude and the directions of uncertainty of estimates based upon these kernels. And you can see that they're changing both the size of the uncertainty as well as the orientation of those uncertainty ellipses. Moreover, by changing this distortion kernel, it actually changes the optimal experiment design. If this were the optimal sampling period for the true data, then this, which is a much faster sampling period, may be optimal under a given distortion. So by keeping track of what the distortion is, helps you to fine tune your experiment design to better account for that distortion so you lose less. And then finally, if we had a limited experiment budget, let's say $1,000, and we can do 2 different experiments at the same time. Let's say we can do fish for $1 per cell, and we can do flow cytometry for 20 cents per cell. The total budget is $1,000. So we'd be able to do any experiment in this triangle. Yet there's an optimal experiment, a non-trivial optimal experiment that uses some cells from experiment 1 and some cells from experiment 2. Uses the same budget, but is able to get the most information. So if you did just fish experiments, you'd get the uncertainty in red. If you did just flow cytometry, you'd get the experimental uncertainty in green, but you can do better. You can get through a mixture, you can find what's the optimal experiment to do. And the conclusion for that is by accounting for errors associated with different experiments, we really can start to maximize the reward for a finite budget by keeping accounts of how the different experiments are distorting the reality in our observations. And so the conclusions are central dogma is noisy, experiments are expensive, they take a lot of time and a lot of effort. There are an infinite number of these experiments that we could potentially do, and we don't have time to do them all. Every choice is going to affect the uncertainty and our conclusions. The Fisher information matrix helps us to estimate the expected uncertainties that we're going to have after doing an experiment, which allows us to select which experiment to do to minimize that uncertainty. The finite state projection is an approach that allows for the computation of the Fisher information for arbitrary non-Gaussian stochastic processes and for any arbitrary corruption of unavoidable probabilistic distortions. And with that, if you would like to learn more, I encourage you, the slides are all available. I encourage you to contact some of these postdocs and students to learn more. If you want to learn more about stochastic gene regulation or about experimental design. And with that, thank all of my lovely collaborators and students and postdocs. Everyone in pink here contributed somewhat to what I've presented here today. All right, and that's all. I'm right on time, but no, happy to take as many questions as you'll let me.
That's fantastic. Thank you so much, Brian. That was really, really interesting. It looks like we've got, maybe first we'll check for any questions in the audience. And it looks like Jake wants to ask a question and Blake wants to ask a question. So go ahead, Jake.
Hi, Brian. Thank you for the talk. I was going to initially ask when you got towards the end about how you said that the one particular model, I think it was the differential equation model, didn't fit as well as the other ones, but it was also using the same Fisher information to come up with it. Is the reason you would keep information like that, sort of like you said at the end, due to if you had budget restrictions, you would keep some of these more error-prone models just to have around, or is there other reasons that you would keep sort of to keep these models that you said you can kind of intuitively tell that there are ones or better ones that you could come up with?
So in that part, when I was talking about the difference between the ODE-based model and the discrete chemical master equation-based model, that was the difference in those two different lines. The ODE-based model has an underlying assumption that noises or measurement noise, or how wrong you do your measurement. The assumption there is that these things are Gaussian, and they're not Gaussian in the kind of processes that we're looking at. So in that case, the one that was wrong, that was giving you the wrong optimal experiment design, that was doing so because it had an improper assumption on the derivation of the Fisher information itself. In the second part, when we're combining different types of experiments, here we are using the correct Fisher information based upon the finite state projection calculation, and we're using the correct probabilistic distortion events. What we're finding here in that case, and the thing that's going on, let's see if I have, yeah, there's a picture here. So if you look at this, maybe I have it here. Okay, if you look at the green line and if you look at the blue line, what you're going to see in the ellipses, you see they have slightly different orientations from each other. They're both losing information about the process. They're both losing some information from the red line, but they're losing different parts of the information. So the optimal combination of experiments is going to be some mixture of these two, because although both experiments are distorted, some distortions affect how well you can learn certain parameter combinations, and other distortions affect how you're going to learn a different set of parameter combinations. And so the optimal experiment turns out to be some combination of those, because one's going to do a little bit better than the other under a certain circumstance. They also have different costs associated with them as well. So if you get as much information from the cheap experiment as you can, and then fill it in with the more expensive experiment, it would be kind of an intuitive way of looking at that.
I see. Okay, that makes sense. Thank you for the clarification.
Fantastic. How about Blake?
Yeah, thanks. That was a great presentation. I was wondering, this is more out of curiosity. You mentioned the viruses, how they have this little trick that they play where they can encode, two proteins at once. And I was wondering, do we know how often it just so happens that you have a chain that can produce two useful proteins with that little offset?
It's been found in a lot of viruses. The one that we're looking at is the HIV Gag-Paul complex. In that case, it's 9% of that particular construct is going to give the 9% are going to give the non-canonical, 91% give the canonical. But this is something that seems to be very prevalent in viral processes. And again, the reason that they want to reduce their genome as much as possible. So if you can use the same length of RNA twice to encode 2 different proteins, that's going to decrease your DNA footprint. And that means our host immune system is going to have to work a little bit harder to find it and to eliminate it.
Gotcha. Perfect. Thank you.
But in terms of what, it seems to be very, very prevalent in viruses that both either using this frame shifting to do that, or using these irises to hijack the machinery so they can insert themselves into regular RNA, but take over take over the ribosome machinery and continue translation. The other thing that the irises do that's really kind of cool and we looked into was under stress, cap translation shuts down. So that our regular normal translation under stress, our RNA are just going to stop translating because we want to deal with the stress, and we'll turn on some other genes perhaps. But the iris does not get shut down. So as a result of stress, the iris actually gets more resources because it has more ribosomes available to it. And it actually maybe even increases a little bit under stress. And we can actually see that and try to model that as well in experiments.
Fantastic. How about Jorge?
Hi, am I coming through?
Yes.
Hi, thank you so much for everything you've presented here. I remember hearing you say that the underlying goal was to try to see if we could have some control or total control over the different dogmatic processes. And I'm wondering ultimately, like, how much control do we have over that already? Or if we're already there at controlling every stage, or is there a measure by how well we can control all these processes?
I think so a lot of folks in the synthetic biology community are really working on ways of controlling. We're getting, I'd say, this is a really rapid direction of ongoing research. And the one place where I'm seeing so much control showing up is optogenetic capabilities. So when we're watching these, or at least at single cell level, you can imagine you can watch a cell, you could shine a light on it. Optogenetic means you shine a light on it, it changes the conformation of a molecule inside the cell. Sometimes it releases a molecule from a membrane, sometimes it activates a molecule, sometimes it causes, you can design it to do what you want to do. Sometimes you could shine a light on it, it causes 2 molecules to become magnetized so that they bind to each other and form an active complex. Using these kinds of models that we've been presenting, you can ask how much light do you need to shine on it when in order to activate gene expression in one cell but not another cell. We have a paper that's just out on the bioRxiv right now, 2 of them actually, well, one, there will be 2 by the end of the week, which hypothesizes that it would be possible to control lots of cells using a single input, but differentially control them by shining a single light on them, and then choose a cell, turn it on, and choose another cell, turn it off, even though you're using literally the same input. And this is something that could only be done in the context of stochastic processes. And so I think this whole about variability, noise, and control, I think this is a pretty open field of research, because what you need to do to control something that's stochastic is very hard. But what you can do to control a process that's stochastic is actually more than what you can do for a deterministic process. And I find that to be a really fascinating future research areas. See what can we control. So, but moving forward in terms of other things we can control, we can change in all the translation experiments I was showing you, we could change the lengths of those genes. We could change the codon usage of those genes by putting in different codon sequences, and we can show that we can use that to adapt the translation dynamics. In that frame shifting example, we don't really fully understand this, but we could kind of control the fraction of cells that are giving 0 and -1 frame by inserting small oligo regions of the frameshift site itself. So if we can inject that into the cells, it seems to stimulate more of them to occur. And then you could increase the amount of frame shifting that occurs inside the cells. So there's a ton of control mechanisms out there. I don't think we fully understand them, but we're able to develop models for them, one at a time. I don't think we have a fully principled way of doing it for everything, at least not in eukaryotic cells. Some of the stuff that folks are doing in the bacterial cells is extremely much more evolved than what we're doing in human cells here.
Thank you so much.
Fantastic. Thank you so much, Brian. Why don't we thank Brian one more time?
Thank you all. Thanks for the great questions, and thanks for having me here virtually. It's a shame I couldn't see any of you.